Okay, let's see. Okay, welcome, chapter 25. So we are going to be talking about bacteria archaea today. Talking about prokaryotic cells. So if we remember back to 1406, or maybe another biology class you took, the prokaryotic cell has no nucleus, no membrane bound organelles. So all life today or in the past can be traced back to a prokaryotic cell that was our common ancestor. And again, these are, I should mention, these are also unicellular, always. So just consisting of one cell, no nucleus, no membrane or bound organelles. Now, if we look at the three domains of life, which are the bacteria, the archaea, and the eukarya, two of these three domains consist of prokaryotes. Both the bacteria and the archaea consist of prokaryotic cells only. And one interesting thing to note as we look at this phylogeny is that the eukarya, the domain that we belong to, shares a common ancestor with prokaryotic archaeans more recently than the bacteria and the archaea share a common ancestor. So that's kind of interesting that we are more closely related to archaeans than archaeans are to bacteria, especially in light of the fact that we used to group them together into a group. We used to call them Kingdom Monera, but recent DNA evidence made it very clear that these two groups of organisms, although both unicellular, both prokaryotic, and to our eyes looking very similar, were actually very genetically diverse. So the last common ancestor of all three domains was probably approximately 3 billion years ago. So, which we could also sometimes you write 3 billion years. When you see this BYBP, that's billion years before present, and this is billion years ago. You might see either notation coming up. So again, three domains of life: bacteria, archaea, and eukarya. But today we'll be talking about the bacteria and the archaea, because today is all about the prokaryotic cell. So let's talk a little bit about the characteristics of prokaryotes. Okay, we've already talked about the fact that they are unicellular, that they don't have a nucleus or membrane-bound organelles, so that's a given. What else do we have going on with the prokaryotes? They divide by a process called binary fission. This is not the same as mitosis, which is what a eukaryotic cell would do. They typically have one circular chromosome, and this is in contrast to eukaryotes that usually have multiple linear chromosomes. Some prokaryotes, so some have an additional smaller DNA loop that we call a plasmid that has a few genes on it. And of course they lack any membrane-bound organelles. So here are just a few images of some prokaryotic cells. So the purple one here, we see it starting to undergo binary fission. Here we see a bacterial cell that has lysed, which means it's kind of burst open, and we can see its chromosome. It is all sort of folded in on itself, so you can't tell, but this would be a circular chromosome. And we've also got a few smaller circular plasmids. So the prokaryotic cells prokaryotes in general have posed a bit of a problem with determining evolutionary relationships for a couple of reasons. First of all, morphological information is limited. Right, we just have fewer structures with which to do comparative anatomy. And this was especially true before, we had the advancements in microscopy that we have. Genetic data has recently been used to classify prokaryotes, and that has been a wealth of information that has helped clear up a lot of misunderstandings in our classification. So genetic data is important, but there are some issues here too that can make figuring out evolutionary relationships among prokaryotes a little bit tricky, and that is that prokaryotes commonly engage in what we call horizontal, also known as lateral, gene transfer. So lateral gene transfer is the transfer of genes within the same generation. So if we think of vertical transmission as the transmission of genes from parent to offspring, right, going from one generation to the next generation, lateral or horizontal gene transfer is within the same generation. And this might be, so it can be among members of same species, but not necessarily. It could be one species to another. So you can probably appreciate how that's going to make things a little bit confusing if we have one species, say, transferring a piece of its genome to another species. When you go to analyze them genetically, you're going to see some genetic similarities, but that similarity is because of horizontal gene transfer, not because of common ancestry. So you can see how it gets very sticky very quickly, and this horizontal gene transfer is common among bacteria and archaea. It happens quite a lot, in fact. So let's talk about how it happens a little bit. So how does this horizontal gene transfer happen? Well, there are three processes that can do it. There is transformation, there is a process called transduction, and a process called conjugation. Okay, so let's talk about transformation first. There we go. So transformation happens when a prokaryote takes in foreign DNA from its surroundings. So we'll say, in this case, that it's a bacteria, so we'll say the prokaryote takes in foreign DNA from its... And so the most common scenario here would be that, in a particular environment, one bacterial cell, say, whoops, sorry about that, lyses and releases fragments of DNA to the environment, and then another living bacteria takes it in. And many cells have cell surface proteins that will recognize DNA from closely related species and allow them to be transported into their cell and actually even incorporate those genes into their chromosome. So when this happens, we have now a recombinant organism. So when you have a recombinant organism, that means it has DNA from two different entities. So that is one way to get it in. The other way to get it in is through a process called transduction. And this process relies on a bacteriophage, which we also sometimes just call a phage, which is a virus that infects a bacteria. So the idea here is that when the phage goes to infect the bacteria, so normally the way this would work is the phage, the virus, would come and inject its DNA into the bacterial cell so that it can hijack the host cell's biological machinery to help make new viral DNA and proteins for it. That's what the virus is going for. But sometimes a mistake happens. And sometimes when the virus goes to make new viruses, instead of packaging viral DNA into the new virus particles that get made, instead a piece of the bacterial chromosome is inserted instead. So see how this virus here, instead of the blue viral DNA, it's got a piece of the red. That's a chunk of the host chromosome. So sometimes that happens inadvertently. And when this virus goes to infect a new host cell, instead of injecting viral DNA, it's injecting a piece of the chromosome from the previous bacterial cell it visited. And this piece of this chromosome can then find areas of the chromosome that it matches up with and actually be incorporated, creating a recombinant cell. So the same, we end up again with a recombinant bacterium, and it's the same effect as before, but just a different method of getting it in there. And finally, our last method of making recombinant DNA is conjugation. So in conjugation, DNA transfer from one prokaryote to another happens through a sex pilus. Which temporarily joins the two members together. So here is a sex pilus forming. It's like a little one of these cells will be creating this sex pilus and joining up to the other cell so that it can actually send a piece of DNA, usually from, for example, a plasmid through, or even it could even send a whole plasmid through and insert it in to the other cell. So another way we can get a recombinant prokaryote made. Okay, so I'm going to stop there for this part of the video. This lecture is kind of a doozy. There's going to be quite a few videos here, so we're going to short. So I'll see you in the next video.